let me take a second to introduce our first AML expert, Dr. Naval Daver. Dr. Naval Daver is an associate professor in the Department of Leukemia at MD Anderson Cancer Center. Dr. Daver is a clinical investigator with a focus on molecular and immune therapies in AML, myelodysplastic syndromes, and myelofibrosis. He is the principal investigator in over 25 ongoing institutional, national, and international clinical trials in these diseases. Dr. Daver has published more than 150 peer-reviewed manuscripts and has authored numerous abstracts at national and international conferences. Our second AML expert is Dr. Uma Bharate. Dr. Uma Bharate is an associate professor in the Division of Hematology at the Ohio State University. She specializes in the treatment of myelodysplastic syndromes, acute myeloid leukemia, myeloproliferative neoplasms, and clonal cytopenias of unknown significance. Dr. Bharate is the principal investigator on numerous investigator-initiated clinical trials, as well as the national lead and site investigator for several MDS and AML studies. Her work has appeared in several peer-reviewed publications. I'd like to thank you both for joining us today. Thank you for taking the time out of your busy schedule today. And I know that our patients and caregivers are thrilled to have you here with us today. And we can't wait to hear what you have to share with us. There will be time for questions, and you can use the Q&A button at the bottom of your screen to submit a question. Okay. So with that, I will let Dr. Daver take it away, and you can share with us your main takeaways from the Eha conference, and then we'll take some questions after you take us through those.
All right, yeah, thank you very much to the HealthWell team for inviting us. Happy to be here with Dr. Bharate and discuss some of the updates from last month's Eha meeting. So, I mean, I don't have slides, but I'm going to kind of go very high level on what I thought were the key takeaways from both Eha and Asco. There were a lot of things that were repeated at those meetings. They're 2 weeks away from each other, so we often present similar data. It's a little bit of a different audience. Asco is more US focused, Eha is more European focused. But really, I think the number one thing, at least for me, from Eha especially, was the frontline data of the what was called the Quantum First study, which is a study of quizartinib in combination with intensive chemotherapy versus placebo-intensive chemotherapy. So just as a background for those joining, Flit3 mutation is a very common mutation in AML. It's seen in about 30 to 40% of patients, especially in younger patients below 65. It's quite frequent. The frequency goes down with age. And this mutation is associated with a more aggressive phenotype, proliferative disease, leukocytosis, high blast. And in the past, when we first published on this, the German group, our group, MRC, about 20 years ago, the outcomes were very poor for FLT3 mutated AML. It was actually probably one of the highest risk groups with 5-year survival of about 20-25% in all of those reports. So since that time, there's been a lot of efforts to start identifying and developing FLT3 inhibitors. One of them that's currently available and approved and has been used for the last 4 years is a drug called midostaurin or Ratify. This was Rydapt, which was approved based on the Ratify study, which was a phase 3 frontline study that looked at midostaurin intensive chemo versus placebo intensive chemo, and the intensive chemo being the standard of care at that time, and showed that the midostaurin intensive chemo improved both response rates, overall survival, and post-transplant outcomes. And we were now starting to get closer to 45-50% long-term survival, much better than 20-25%, with both midostaurin and transplant being incorporated to treat FLT3 AML. But even with that improvement, a number of patients relapsed. Midostaurin is a multi-kinase inhibitor. It was not really developed specific to FLT3. That happened to be one of the targets. It has side effects, including nausea, GI, taste issues. And so there has been a search in the last 8-10 years for more specific drugs that were designed and developed in the lab to be FLT3 inhibitors specifically. And these are 2 drugs now that we're quite excited about, gilteritinib and quizartinib. Gilteritinib already has been approved few years ago in the relapsed AML setting, where it's proven to be probably the best agent for FLT3 mutated AML, and especially now being combined with venetoclax and chemo, continues to improve response rates and gives us hope for relapsed FLT3 mutated AML patients where we had no good options 5, 6 years ago. But the best outcomes usually are by putting our optimal therapies in the frontline setting. That's something we have learned historically from ALL, from myeloma, from lymphoma. And so now we have started moving the second generation FLT3 inhibitors. These are drugs like gilteritinib and quizartinib to the frontline setting, where we are actually hoping to cure a good proportion of patients right from the beginning with optimal therapy. So this is where quizartinib has been combined with intensive chemo, and this was studied in a large 500 patient randomized study called the Quantum First, and quizartinib intensive chemo versus placebo intensive chemo. The output here was overall survival, and secondary endpoints were event-free and CR/CRI. And basically, the summary is that quizartinib very clearly improved the overall survival in the ITD population. Now, one of the differences in this study compared to the ratify midostaurin study was that this study allowed patients of all the ages from 18 to 75, whereas the ratify study only was 18 to 60. And in fact, 40% of the patients in the quantum first were greater than 60 years, which is, of course, a more difficult population with survival not being as high as the less than 60. And so that data of the split between less than 60 and above 60 was not shown, but it will be shown in the near future. And I think looking at all of that, to me, quizartinib looks like it is going to be a very important drug and probably the one that we will be preferring in the future, especially for ITD. Now, there is another type of FLT3 mutation called the TKD mutation. And in those patients, quizartinib does not work because it's a type 2 FLT3 inhibitor, which only works for that ITD, which is the common one, 80% of all FLT3, but it's not the only one. So if we find a TKD, then we would still be considering either midostaurin or maybe in the future gilteritinib, which is also being evaluated in frontline treatment. So this is probably going to be the one study that clearly, assuming and hoping that the drug will get FDA approval, which we think it will based on all the data, could become a standard of care shifting study, and we're waiting for more updates in the publication, et cetera, to come out. Other than that, I think, some of the other interesting data that was shown is in the TP53 field. So that's something that we have been working on. So there's a lot of efforts like FLT3 to start seeing what we can do to improve the outcomes of TP53, which remains a very difficult subset with very poor outcomes. We had a drug called APR, which unfortunately, in spite of improving the CR rate, did not meet the statistical endpoint that the study had set forth. And so that study went down as being negative. And we don't know if the APR will be continued to develop, although I think it is a good drug, maybe not only in TP53, but also potentially non-TP53, because we're now learning it has other mechanisms of actions, including ferroptosis and working through the redox pathways. So maybe there's a way to get this drug back. But in the meanwhile, we have shifted more and more of our attention towards a group of immune therapies called the CD47 SERP alpha immune pathway. And so we've historically used T cell based immunotherapies in solid tumors and lymphoma now for the last 15 years. And it's probably been the greatest breakthrough in treatment of solid cancers and lymphoma in the last 50 years. These are called immune checkpoint drugs, but they work on the T cells to remove the block. So cancer is very smart. It actually finds a way to block your body's own immune T cells through receptors like PD-1, CTLA-4, TIM3, and others. And this way, then the T cells cannot fight against the tumor or the foreign antigen as they normally should do. When we get infection, when we get anything foreign body, the T cells are the ones that eradicate it from our body, but the cancer actually shuts them down. So then we and these researchers design antibodies that blocked that interaction between the cancer and the T cells by bringing PD-1 antibodies, CTLA-4 antibodies, and by blocking that inhibitory interaction, the T cells were again free and unleashed to attack the cancer cells. So this is the story of immune checkpoints. Many of them have been approved. There have been more than 25 indications in the last 2 decades. And for many solid tumors, this has been the biggest breakthrough ever. Now, we tried these in AML MDS, and unfortunately, we were not able to get similar degree of successes. There were some subsets, post-transplant, potentially people with extramedullary disease, where we saw benefit, but for the broad AML, MDS, we did not find a great benefit of the traditional PD-1, PD-L1, CTLA-4 sort of immune checkpoints. However, in the process, a lot of immunotherapy work has been going on in AML, looking at different pathways, looking at different ways to leverage the immune system against AML. Remember, stem cell transplant that we have done for 30 years is really an immune modality where we're replacing the defunct, dysfunctional immune system with a healthy donor immune system to continue to eradicate AML. So we know immunotherapy works. It's just, can we deliver that similar approach outside of a full intensive stem cell approach? And so one of the pathways that the group at Stanford, Ravi Majeti, Herb Wiseman, started working on about 12, 14 years ago, and recently have collaborated with Jim Allison and others here at MD Anderson, has been a macrophage immune pathway. This is called the CD47 SIRF alpha. And here what we're trying to do is to try to unleash macrophages, which are also very powerful anti-tumor, anti-infection cells that also the tumor shuts down. But the pathway is the different pathway called CD47 SIRF alpha. And so by blocking that interaction, the CD47 antibodies again remove the inhibition on the macrophages. The macrophages are unleashed, so to say, and then can attack tumor cells. In fact, AML and diffuse large B-cell follicular lymphomas were found to be the two tumors where the CD47 pathway was most highly expressed and macrophage dysfunction was most clear, and higher expression of CD47 actually was shown to be associated with inferior survival. All of this leading to development of a drug. This the first one has been called magrolimab, which is a CD47 antibody, which actually was developed through collaborations of Stanford with a company called 47. And this drug now has shown data in lymphoma, which is published in the New England Journal a few years ago, and also now is being evaluated extensively in frontline MDS AML. And at the Eha meeting, we showed both the data sets. One was frontline MDS, high risk, that Dr. David Salman, my colleague from Moffitt, presented, and the other was TP53 mutated AML frontline that I presented. And I think overall what we're seeing is in MDS, we're seeing very good tolerability, low early mortality, high response rates, and the survival is looking quite encouraging. Median survival not reached now in the frontline higher risk patients, and even in TP53 mutated MDS, where historically we've had 10 to 12 months survival, including with drugs like APR, we're starting to see 16 months. So single arm, we have to confirm this. Randomized studies are ongoing, but there's definite optimism for this in MDS. And in AML, what we found was that there was a specific group of patients, the TP53, where this drug seemed to have a preferential activity compared to what has been seen with the established therapies in TP53, like azavenetoclax, azacitidine intensive chemo. So in that population, we're seeing encouraging response rates, about 50%. The true CR rate still remains around 35%, not as high as we would like, but it's still reasonable in this population, and a survival of close to 11 months, which doesn't sound long, but if you look at all the data with azavenetoclax or intensive chemo, historically it's been about 6 to 7 months. So we think this could be kind of a first building block to get some improvement in TP53 ML. We know that we're still very far away from where we want to be, like in the FLT3 and IDH, and we're probably going to have to build on it with immunotherapies. TIM3 antibodies are starting to show some activity, and maybe bispecifics and other approaches. But I think for the next 5 to 8 years, TP53 is going to be a major area of focus in AML, and is really something we need to find a solution for, because currently a lot of our patients who have that will have very poor survival, and short of getting them to stem cell, we have limited efficacious options. So I think that's the space to keep looking out for, hopefully more data at ASH and then coming next year. The other, I think, last one that I would probably mention is similar to FLT3 and TP53, we're also doing IDH targeted therapy. So there was data that was presented from our group. One of our fellows, Curtis Lakowitz, presented data in collaboration with Dr. Courtney DiNardo from our group on using combinations to target IDH. So one of the big areas of discussion, debate, and interest in AML is how to optimally combine or sequence new drugs. We've been very fortunate through a lot of efforts of people before us who did a lot of the sequencing, biological work to understand targets. We've got multiple drugs approved, 9 drugs to be specific in the last 4 years, compared to only 2 drugs in the previous 40 years. And this is great, but it also now provides opportunity to try to find ways to use these drugs not a single agents, because in no heme malignancy have we ever seen single agents cure patients, but in some form of either combination or sequence. I think this will now lead to many, many trials. Some are already ongoing, others are starting, of looking at different ways, combining drugs upfront, targeted therapies with venetoclax or HMA venetoclax, sequencing them, start with venetoclax, bring in a targeted therapy at the time of MRD, or wait for relapse and then use the targeted drug. These are all big questions. We don't yet know the right answers. At MD Anderson, we've kind of taken the path to combine agents based on how we did it in ALL, where we combined the antibodies and the chemo. And so we have studies of AZA venetoclax with IDH inhibitor, for example, for IDH mutated AML. And the data that was shown at ASCO was that this combination seemed to be, number one, quite well tolerated. We did not see any unexpected or concerning significant toxicity beyond what you see with AZA venetoclax or AZA IDH. And again, at least on the data that we had, which is limited and single arm, the response rates looked very, very encouraging with MRD clearance and molecular clearance and good durability. So we think for IDH, there could be a argument to be made to use these drugs upfront. Of course, we need to see long-term survival and then compare it with data that's been presented and published with AZA-VEN IDH. But I think that this could be an interesting path forward. In parallel, there will be studies, I'm sure, looking at AZA-VEN sequenced with AZA-IDH or with a IDH brought in later. And then we have to see what will be the best approach. But that data was quite encouraging moving forward. So I think there are many other things, but these are kind of the top level, things that I thought were of interest to me. And maybe here I can turn it to Dr. Bharate for her thoughts and comments.
Thank you, Dr. Davar. I don't know, Carrot, if you want to do a Q&A right now based on your agenda, or you want me to go?
Yes, I think we can probably look at the Q&A and take a few questions based on Dr. Davar's comments, and then we can go from there. Let's see. Okay, we have a question here that says, what clinical trials do they have for AML lymphoma Flit 3? That is very specific. I don't know if you guys have an answer for that, but I'm gonna go through
here. Is it just coming to you?
This is for AML lymphoma Flit 3.
I'm not sure what AML lymphoma. I don't know if they're talking about like a biphenotypic leukemia with like AML with the lymphoid component. We see that sometimes. It's about 2 to 3% of AML. They can have a FLT3. It's quite a rare disease, as you said. Yeah, maybe we see like 3 or 4 of those FLT3 mutated biphenotypic in a year or 2 years, even at big centers.
Oh, wait, I think she corrected herself and said
Okay.
AML leukemia Flit 3. What clinical trials do they have for
Right.
AML leukemia Flit 3? So there's probably quite a lot.
Yeah, there are a lot. I mean, I can just mention that, there are trials with gilteritinib, there are trials with quizartinib, there's trials with the new FLT3 inhibitor, crenolanib, not that new, but it's been around for a couple of years, but it's a good option for people who have failed gilteritinib, quizartinib. There's even new generation FLT3 inhibitors like HM432 and CG806. So bottom line is there's a lot of trials. It really depends on frontline versus salvage, age, performance status, fitness. A lot of this information is available on the LLS leukemia lymphoma site, as well as on the MD Anderson clinicaltrials.gov. Ohio, I'm sure, has similar ones. So, feel free to reach out either through your doctor to Dr. Bharate or me, or directly to us, and we're happy to kind of discuss it more offline. Uma, any thoughts?
Now, I agree with that answer. I think, as you said, there's a lot, and it depends on where you're geographically located, where is the nearest center to you, and what they have available. And the resources that Dr. Davar mentioned are really good ones to explore that. But really talking to your treating physician and asking them, what the best place to refer you or your loved one is, just to explore these trials.
Okay. Okay, there's one about Should I go to a specialist hospital for AML leukemia, Flit 3? Or what should I ask the doctor at the hospital if they specialize in AML? What kind of proof do I need to know that my son is in the best facility? I think this would be for pediatric AML.
You want to try, Uma, or yeah.
Sure. I think that's a really good question. I think in general, for any AML, whether it's an adult or a child, you really want to be at a center, a medical center that treats a lot of these patients. So if they say that they probably see 1 or 2 or even less than 10 or less than 20 AML patients a year, that's probably not somewhere that, because they don't see a lot of volume, they may not have as much available in terms of clinical trials or different things that AML patients need. I think that is a good question to ask your son's treating physician. And usually in children or pediatric cases, most of the centers that treat AML are pretty big. And there's specialized children hospitals, and the oncologists there are very specialized in these type of blood cancers. However, I think it's always a good question to ask and ask if maybe another center or a bigger center has more options for treatment, more clinical trials, and if they feel like that would be a better option for your son. However, I think the big thing to remember is AML is an aggressive cancer and patients need treatment pretty quickly. So what we don't want to do necessarily is to wait too long to treat somebody. And so I think you have to balance, again, where you're located, what treatment your son is already getting, and how to make sure that his treatment team is working with everybody else to make sure he gets the best treatment possible.
Thank you, Dr. Bharate. I think this is a good general question. What are some of the obstacles to progress in AML? A difficult one, but
yeah, I can start with that. I personally think, this is an area of big debate, and I know a few centers think different from others in that. I personally feel that large randomized studies are very difficult and are gonna lock us up in long delays. We've seen this with midostaurin. We've seen it with Quantum First, which in fact was started so long ago that it was started before the current standard of care. And so the big debate with that study is, even though I've seen the data and we know the FLT3 field and we believe it's better, it's just going to be hard because it started 7 years ago and then midostaurin came along meanwhile, and now we have a study that's randomized against placebo, and so it becomes hard. So I think more and more, like myeloma, we're going to have to switch to studies that are either using non-survival endpoints, so using either MRD, PFS, as well as not full-blown long-term phase 3s, but phase 2s with kind of a focused population, randomized rapidly, done in good centers. I think personally, now that we have the benefit of multiple drugs, we need to find quickly ways to use them, whether it's combination sequencing, other ways to do them. That may be, I think, the biggest hurdle. The FDA, actually, I have to say, has been extremely supportive and has identified both MDS and AML as major areas of unmet need, and are very supportive and helping to look at alternate endpoints, to look at novel ways to conduct the phase 1 studies, to move to combinations quickly, if a drug is likely to have benefit only in the combination setting. But yeah, I think just avoiding very long randomized studies. I think the other big hurdle is, of course, trial enrollment itself. We still see only about 5%, in the country, 3 to 5% are enrolled on clinical trials. So if we were even able to get 25%, which is still not great, but it's good, we would have 5 times quicker completion of the trials. And so a number of these studies, like magrolimab, like NK cells, like Menin Inhibitors, we could have already finished and hopefully even gotten them on the market. So, and of course, that would mean thousands of patients would have had access to them earlier. So I think those, to me, are kind of the two biggest hurdles. We're definitely better than we were 6-7 years ago, but I think we can continue to improve on this.
And on that front, on the clinical trial front, we that's something that we try to talk about a lot here at HealthTree, and we do the podcast that we really try to focus on clinical trials just to spread awareness. We also have the clinical trial finder on our website where because, clinicaltrials.gov is very hard to navigate. Do you find that it's a communication factor in that trials are not being communicated by doctors more so? Patients are not inquiring about them? Is it a combination of both? What do you think is the issue there?
I think it's a combination of both things. I think, as you know, your previous question alluded to, if you go to a center that doesn't see a lot of acute myeloid leukemia, one, they may not have the trials and they may feel this urgency to start some sort of treatment, which then very often precludes a patient from starting on a trial because you really want to start the trial, in the beginning when you have had no treatment for the disease so you can get on the appropriate study. And then two, it's hard because patients don't necessarily always know to ask that question. Is there a clinical trial available? If not at that particular hospital, can they be referred or recommended, hopefully somewhere nearby? Where there is a clinical trial available that's appropriate for themselves or their loved ones. So I think it's both. And then thirdly, you alluded to this where it's just hard to navigate to find that information. And Dr. Davar mentioned the Leukemia Lymphoma Society, that's one resource. You guys are a resource. Your website is a resource. And I think really talking to the treating team at your particular location to say, hey, can you tell us more about these trials? Is this appropriate for us? Should we go there? As far as I know, all of us are a pretty tight-knit community, and any of us would be happy to facilitate, that type of discussion, all across the country, really.
We've also seen in some of our Facebook groups that there's a good bit of misinformation out there about clinical trials and what's involved, and being concerned that they're getting placebos or in some cases no treatment, or so I think it all stems from the education piece. So
yeah, I want to make a point on that actually, because it's actually not that complicated if one understands, the regulatory. Just to make it simple, it's common sense. So we would never do a trial that is not going to move the field forward, right? So it's 100% given that if you're going on a trial, it's going to already be geared to improve on the standard of care. There would be no motivation if one wants to use any argument, scientific, academic, financial, to the companies, FDA, regulatory, right? The only reason one would, because trials take a huge amount of time and a huge amount of effort, manpower. So the only reason for one to do them would be to make sure that it's going to move the field forward. So even if you're going to get a placebo in the trial, it's going to be a placebo added to standard of care. So if you're going to get your treatment in a small community hospital, wherever you are, the trial will randomize you to that same treatment or something with that treatment plus drug X. So the real scenario is either you're going to get what you were going to get anyways, or you have a 50% chance of getting that plus something else, which all the scientists and the experts believe could make it better. So there's a no loss situation. There's no trial we will ever do where we're going to say we're going to give you something less than standard of care versus the drug, because that trial has no future. If it's positive, people will say, well, I don't care because it didn't even beat the current standard of care. So why would the community doctors and the FDA and others use it? So I think that the confusion, I don't know why. I think the word trial maybe makes people confused. Maybe so we usually call it clinical investigation or clinical protocols, because the trial here is not just nothing versus something. It's the current standard of care that everybody's doing versus something that hopefully will make it better. I think this is a very important point.
And to add to Dr. Davar's point, because he mentioned in his discussion, sometimes the standard of care changes and the trial may not change. So for example, if the standard of care is X plus Y, and the trial is just X plus something else, and the standard of care has now changed, I think that's an important question to ask the person that is educating you about the trial. Will I at least get the current standard of care? And so then if they're explaining the study well to you, they will say, absolutely, this is the current standard of care, and you may either get that extra drug that Dr. Davar mentioned, or you may get nothing, or you may get a placebo. So at least you're reassuring yourself or for your loved one that the current standard of care is being given to you no matter what arm you get assigned to, that minimum, wherever you are, that will be given to you. And if that is the answer, then I think I would personally feel very comfortable enrolling myself or my family on such a trial.
Yeah, that's a very good point. I agree with Dr. Bharate. Yeah.
Yes, thank you guys for clarifying that for us. I think that's great information. I have like 2 more questions that I think, and then we can move on to Dr. Bharate. What about the MLL mutation? What are we seeing for trials for this? Anything as maintenance after a stem cell transplant?
I can start. I mean, so, this is a very, I didn't mention it because there wasn't much data at Eha and Asco, but all of us, Dr. Bharate, me, and most of the AML experts are very excited about this group of drugs called Menin Inhibitors. This is probably, we think now, the third targeted therapy after Flit3 and IDH that will be established for AML, and it works in a group of mutations or chromosome abnormalities called NPM1 or MLL rearranged. And there are a few other subsets where it can work as well. In addition to that, we're learning a lot about the biology. What's very interesting is that this group of drugs is not like the FLT3 inhibitors. It doesn't directly work by blocking receptor and leading to cell death. And it's not like chemotherapy that kills the cell. So it really works by promoting differentiation. So what happens normally is when you have AML, there are mutations that accumulate, and the leukemia stem cells or the blood stem cells cannot go through the normal progression of stem cell to pluripotent cell to monocyte to neutrophil. So, and then the neutrophils eventually die on their own after protecting the body from infection. So there's a differentiation blockade at some point, and all the cells get stuck there, and then over time they multiply and lead to leukemia. So one of the pathways that was identified through a lot of research that a lot of experts for the last decade have been worked on is the Menin pathway. And so this pathway, when you remove the overexpression of certain enzymes like MEIS1 HOXA, then you start getting normal differentiation. So all those cells that were stuck in an early, premature, abnormal leukemic stage start maturing to normal neutrophils and monocytes and healthy cells. And over time, you actually have all normal cells. So it's actually a very, normal way of approaching leukemia rather than killing it, to actually let it mature into the normal healthy cells, which is good because you don't see all the side effects you will see with chemo, like nausea and diarrhea and mucositis. So of the many, there's about 6 or 7 trials ongoing. The 2 that have been the most advanced are Syndax and Cura, which have shown data at the ASH meetings last year. And we see somewhere between 40 to 50% remission rates in multiply relapsed third, fourth salvage patients with single agent. So this is very, very clear activity. It's not something we would see normally with any single agent. And so we believe that this group of drugs will have a future. Now, again, I think even though it has good responses as a single agent, we're still at 40, 45%. Duration is only about 6 to 8 months as far as we're seeing, but eventually it'll move into combination in the front line. And those studies are already being designed with combinations of venetoclax, with HMA venetoclax, intensive chemo, etc. Post-transplant, there's no trial that I'm aware of. I know there is a upcoming trial in the near future that's going to specifically look at post-transplant maintenance with Syndax. There is a way to get the Syndax drug through compassionate use. So if you're a treating physician, is it a larger academic center, they could approach the company and ask for compassionate use. We've had 5 or 6 patients now who have gotten compassionate use Syndax post-transplant. I personally think it is actually makes a lot of sense, and I would strongly recommend to try to do it, because that group of MLL rearranged is very high risk. And so even if you go to transplant, we still know there's about a 50 to 60% rate of relapse in the next 2 to 3 years potentially. And if we could do a maintenance menin inhibitor, maybe we can cut this down to 20, 30%. So I think it makes sense to try to get that if you can.
Okay, thank you, Dr. Dauber. And then our last question is, just find this one. Do you expect the prognosis for the elderly AML patient to improve?
I can start with that. I think there is a huge movement in the AML community to focus specifically on what is termed elderly AML, though I know there's several of my patients that would take a little bit of offense to that term, because in our community, we are defining older AML as patients over 60. As Dr. Davar mentioned, many of the studies were either over 60 or under 60, and that's sort of the cutoff. But I think realistically, we all know that AML is diagnosed in patients, usually over the age of 65. Most of our patients are in their 70s. And so a lot of trials are really focusing on that age group and really looking to move the field forward where we can design different regimens that minimize toxicity and maximize efficacy. I think Dr. Davar mentioned multiple combination trials looking at these type of agents so that we can target mutations specifically and improve outcomes in older AML patients. So I do think there's a lot of promise, there's a lot of focus, there's a lot of resources being devoted to older AML patients. And I personally am quite optimistic that we're going to continue to move the field forward.
Great. Thank you so much.